Hi everyone, my name is Jenny Lynn and my partner is Rebecca Wang. Today we're going to talk about conjugation in bacteria. Here we have a diagram of how bacteria looks and the main components of bacteria. Today we'll be mainly focusing on the plasmid. So, what is conjugation? Conjugation is mediated by a particular kind of circular DNA called plasmid, which replicates independently of the chromosomes. It involves a donor cell and a recipient cell, in which the process does not create new bacteria but allows them to share antibiotic resistant genes. The main structure in these plasmids is the bridge like structure that connects the two cells. How does the transfer of antibiotics occur? Well, first the plasmid carries the antibiotic resistant gene. The donor cell then extends a pilus to attach to the recipient cell. The pilus pulls the cells closer to form a mating bridge. The donor then makes a copy of the plasmid DNA, and the copy travels through the pilus to the recipient bacteria. Now, the recipient bacteria can also be a donor and spread the antibiotic resistant gene. We see this during our conjugation lab. Strain 1 was resistant to streptomycin, while strain 2 was resistant to ampicillin. Strain 1 was able to grow on the LB-STR plate because it was resistant, while strain 2 was able to grow on the AMP plate. After making a mating plate, where we mix strain 1 and strain 2, that bacteria was able to grow on both the LBSTR, LBAMP, and also the LBSTR plus AMT because the bacteria shared the antibiotic resistant genes, making it resistant to all of the plates. The type of genes transferred during conjugation are usually antibiotic resistant genes that help bacteria survive drugs designed to kill them, metabolic genes that allows bacteria to digest new nutritions or survive in harsh environments, virulence genes to help bacteria resist in immune systems, and toxins so that bacteria can cooperate with other microorganisms. Now I'm going to pass it on to Rebecca.
Hi everyone, I'm going to do a quick sum up what we said before. So bacterial conjugation, which is basically how bacteria share genes with each other. So think of it like passing along a cheat code that helps them survive and adapt. Next, why is conjugation important? Conjugation is important because it allows bacteria to share genetic material directly with each other. This increases their genetic diversity and helps them adapt to new environments. During conjugation, a donor bacterium transfers a plasmid, which is a small circular piece of DNA, to a recipient cell using a pilus, which is a bridge-like structure. This process spreads useful genes such as antibiotic resistance or toxin production, which makes bacteria stronger and harder to kill. Overall, conjugation plays a major role in bacterial evolution and survival. Next, how it spreads through a population. So once a bacterium gets a plasmid, it can pass it on to others, creating a chain reaction. In big colonies of bacteria, this can happen super fast, so the entire population can gain new abilities quickly. This rapid gene sharing is one reason antibiotic resistance spreads so quickly and why bacteria are so tough to fight. So a summary of our whole presentation, conjugation is a powerful gene sharing system. It helps bacteria survive, evolve, and adapt, but it also creates real medical challenges. Understanding it is the key to fighting bacterial infections and controlling antibiotic resistance. And that's it. Thank you for listening.